move on to our next presenter, and that is going to be Dr. Tim Brenneman. Dr. Brenneman received his BS in biology from Goshen College and his PhD in plant pathology from Virginia Tech. He joined the faculty of the University of Georgia in 1986 and currently serves as a professor of plant pathology with both research and extension programming in peanuts and pecan as far as his crops that he covers. He has published over 150 refereed journal articles. He's a fellow and past president of APRES and also a fellow of the American Phytopathological Society. Dr. Brenneman's talk is going to be dealing with Aspergillus in peanut seed. An old enemy learned some new tricks. So, Dr. Brenneman, it looks like you've got yours pulled up, so you can take over.
Okay, Johnny, thank you very much. It is a pleasure to be here. I've enjoyed the previous speakers and I've learned a lot already. My task today is to give a little background on the ecology and biology of Aspergillus, and also then give an update on some new developments in the seed treatment arena, the fight that we've had with this fungus, getting good peanut seed. So that is my objective. I did, as I said here, I titled this An Old Enemy Learns Some New Tricks. And this is indeed an old enemy. Unfortunately, my screen is not advancing for me. Let's see here. Okay, there we go. All right, we go back to 1729. That's when this fungus was first identified by Micheli, an Italian priest and biologist, the father of mycology. So this is one of the earlier fungi even identified. At that time, we had just, the first microscope had been recently discovered. It was 130 years before we even really had demonstrated that microbes could cause plant disease. Anton de Berry did that. So early times, this fungus was one of the first identified. Micheli, being a priest, looked at the conidiophore, which I've got here in the picture, and immediately reminded him of an aspergillum, a holy water sprinkler. And so that's where the name of the genus came from, Aspergillus, and of course is still known as that today. Some unique aspects with this pathogen: it can grow in very high osmotic pressures, it's very low water potentials, and in very low nutrient levels. So that gives it the ability to grow places some other fungi can't. One would be, for example, mold in buildings. There's a lot of money made these days on remediating mold in buildings, and quite often a significant component of that is Aspergillus, because it can grow under those adverse conditions. Another aspect of this pathogen is it can be a human pathogen. The optimum temperature for A. flavus is 37 degrees centigrade. If we were in person, I would have a show of hands of people who could convert that readily to Fahrenheit. But my calculator tells me it's 98.6 Fahrenheit. So that sounds like a pretty good temperature for a human pathogen to operate at. So A. flavus and A. niger are the second and third leading causes of aspergillosis. Aspergillosis is a very serious invasive infection of humans. It can often be fatal in immune-compromised individuals. The picture there on the left shows Aspergillus growing through human lung tissue, which obviously is a devastating condition. The number one cause of that is actually Aspergillus fumigatus. This species is similar, but it's not a peanut pathogen, but it does occur in peanut fields and in peanut debris. If you have piles of peanut hay that heat up, it would be very common to have Aspergillus fumigatus in there. And one of the hot issues with this is that they're finding a lot of azole resistance in patients that are being infected with this fungus. And it turns out that the strains of fumigatus are azole resistant before they're even treated in the hospital. This obviously is a significant issue. And of course, the potential link back to azole use in the field, because triazoles are one of the main chemistries we use in plant disease management. And that exact same chemistry or very similar chemistries is being used in human medicine. The species groups or complexes we deal with mainly in peanut. I'm just going to refer to these as the A. flavus group and the A. niger group. A. flavus coming from Latin for yellow or auburn. I saw that in one source, had to put that in there for my friends across the river. This one is obviously very well known as a major source of the highly carcinogenic metabolites known as aflatoxins, some of the most potent carcinogens in nature. This one is not as well recognized as a peanut pathogen, however. We focus on it mainly in terms of the aflatoxin production. A. niger is the black-spored Aspergilli. One interesting little tidbit on those is they are a source of industrial enzymes. And in fact, most of the world's citric acid production comes from fermentation with Aspergillus niger. And it, on the other hand, then is very well known also as a peanut pathogen. Both these species are readily seed-borne in peanut. There is some evidence for antagonism, which I guess would not be too unusual with species that occupy a relatively similar ecological niche. The left here is the Aspergillus flavus, and on the right, the big black colony is Aspergillus niger. This shows what it looks like in the field. Aspergillus crown rot occurs usually after the peanuts have emerged, maybe 20 to 40 days after emergence or after planting. At that time, the plants get big enough that they are pretty much resistant, although some can continue to die, especially if you get insect injury. But it's very dramatic because as a picture on the left, the peanuts come up and they start growing robustly, and very suddenly, in a matter of a day or two, can just wilt and go from looking great to essentially being dead. And when you dig those up, like you see on the right, you see the black masses of spores at the basically where the cotyledons are attached just below the soil. I mentioned this is an old pathogen, and it's something you can go back in the literature well before the 60s. This was just one example from Georgia back in the early 60s from Curtis Jackson. He described being in 3 fields where he was finding 30 to 50% stand reductions. Obviously, that would have been pretty devastating to witness. And he described even larger, more extensive, through 24 counties, extensive seedling deaths, stand reductions of 10 to 30% were common. This can be a serious, serious pathogen if we don't have the tools to control it. And another interesting point he made, though, he said that seed lots with greater than 10% A. niger infection were not common. And I was a little bit surprised to see that. Because I've been looking at some seed lots over the past numbers of years. It's not something I've spent a huge amount of my time on, but I do some seed treatment work, and so I've evaluated these each year. And I often will see seed lots with greater than 50% A. niger. And some Aspergillus flavus, of course, but usually those incidences have been much lower. So today we still have issues. This picture is a field I took in Berrien County, not too far from where I'm sitting here in Tifton at the moment. The front part of that field towards the right was planted with farmer saved seed that had a lot of Aspergillus in it. And then he ran out of that and went and got some commercial seed. Obviously a big difference. And we see that a lot, that it's often associated with specific seed lots or sometimes specific fields where there's high populations of Aspergillus. The big frontline defense we have is our seed treatments in dealing with this problem. And we really need those. And one reason is we really have no post-emergence remediation. Once you get to this point, and especially if you're well into the season, 40 days after planting, it's getting very late to replant, and it's hard to come back from that when you have that degree of a problem. With that as a bit of introduction, I'd like to bring you up to speed with what we have seen this spring. 2020 brought some new challenges. In February, our state seed lab reported issues with low germination. They look at thousands of seed lots from across the Southeast, and these are commercial seed from all kinds of seedsmen. And the other thing they were seeing was a high incidence of A. flavus, not A. niger, which is the one we've been focusing on. And this is a picture that they sent me from the seed lab here on the left. You can see the A. flavus growing rampantly, and these are fungicide-treated seed. And the other thing they were observing was a big difference in seed treatments. The Dynasty, which is our standard commercial treatment, was having a lot higher incidence of the Aspergillus, it seemed like, and having lower germinations. Now, this was in some seed lots, not all seed lots, but there were some. I knew of one that was over 60% difference in germination between Rancona and Dynasty, which is a huge difference, especially if you're trying to get commercial seed to germinate at certain levels so you can sell it. For those of you not familiar as much with our seed treatments we use, I put this slide in here to kind of fill you in. We have two. Thankfully, we have two. For a number of years, we pretty much just had one, which was Dynasty. PD stands for peanut dust. These are applied as a dust to the seed at about 4 ounces per 100 pound. Because of the range of pathogens that can infect seed, we have different AIs, active ingredients. Dynasty has a group 11 and a group 12. The ones in red are mainly for the oomycetes that don't really impact our discussion here today. But keep in mind the group 11 there, azoxystrobin, which is a QOI, and that's going to play back into our discussion here later on. The newer seed treatment that's used to a much lesser degree, we've been testing it about 5 years now, is Rancona. And you can see it has a group 3, a group 7, as well as a group 4 for oomycetes. So it has a completely different chemistry mix. And the other thing we can do then to remediate these seedling diseases is to put a fungicide in furrow. So at the bottom of the screen, I've listed the main in-furrow products we use, and I've underlined the one there, Abound. It is a group 11, a QOI, and in fact, it has the same active ingredient as in Dynasty, which is azoxystrobin. So it's the very same active ingredient that we've been using in furrow as our front line of defense for Aspergillus, and that's what we also have in Dynasty. Well, to try to understand what was going on, I got some seed lots and started plating them out. We simply treat the seed, put them on a general growth media like potato dextrose agar, and it became immediately evident in looking at some of these seed lots that we were dealing with much higher levels of Aspergillus flavus on the Dynasty treated seed versus the Rancona. These are the same seed right here. On the right, you see there's some things growing on the Rancona. These are generally more saprophytic organisms. There's a lot of things. These were not sterilized in any way. There's some penicillium. There's some other things that are largely non-pathogenic. But a lot of these seed on the Dynasty had Aspergillus, and it was not a niger, and it was a night and day difference between the seed treatments. This is some data from the first 8 seed lots I looked at. These were commercial seed lots from different seedsmen in the Southeast. This shows the percent of these seed contaminated with Aspergillus flavus. And you can see the Dynasty is in the yellow, the Rancona is in the black. The range was 34 to 92% on the Dynasty. The average was 70% versus 0 to 5% in the Rancona. As I said, this was a dramatic difference between these seed treatments. I was very impressed with Diane's slides there, the state-of-the-art labs and equipment they had in Florida. I thought I would show you my state-of-the-art lab in Georgia. If it looks a lot like a ping pong table, it's because it is. And this broke loose in February and March when we were told to basically leave the university. So I plated a lot of these seed and put them in containers and brought them home and sat there and watched the cardinals on my bird feeder while I counted Aspergillus. If you were willing to not play ping pong for a while, you could leave these plates there and you could even evaluate them then for germination. You can get seedling or taproot development, hypocotyls, plumule development. They're on the media. Of course, everything that's happening here is seed-borne because that's the only inoculum in that plate is what's coming from the seed. And you can see how it affects the plants as they develop. And I took some data on that from these seed lots. This shows the percent of those seed that were able to actually form a root, a primary root in vitro. And you can see with the Rancona, that was nearly 75%, and with the Dynasty, it was about half of that. So we're having a very dramatic effect on the seed as they were germinating as well. I had not looked as closely at A. flavus as a seedling or seed pathogen. And so I made a point to do that this year as we started planting some of these seed. And I just wanted to share that so we can maybe help folks recognize. It is primarily a pre-emergence rot. The seed on the left are just several days, less than a week after planting. This rot can occur very quickly. And I will say that's not, I don't attribute all of this to Aspergillus flavus, rhizopus. In some cases, we had some heavy rainfall with some pythium contributed, and that rot can occur very quickly. But a fair number of these seed, and you can see, I think, particularly on the center when there some sporulation of A. flavus, I'm sure it was contributing to this seed rot, as well as then a little later, once they germinated and started coming up. To the right, you can see seedlings in different stages of development. The one on the left is pretty much a healthy seedling. And then we're getting necrosis and lesions at the base of the hypocotyl, like you see in the middle there, as well as the cotyledons, the developing plumule would just be melting down. And we would plate those tissues out, and a very high percentage of those gave us A. flavus, indicating that was the pathogen on those seedlings that were being killed. So what was the result of all this? For one thing, we had a dramatic shift in our seed treatments that were used in the industry in Georgia this year. In February, when this broke out, the great majority of the seed were planning to be treated with Dynasty. UPL is a company that produces Rancona down in Valdosta. A much smaller degree of production. They weren't expecting this. They ran extra shifts to produce as much Rancona as they could. There was a seed that was expected to be good that did not pass germ. And another result was that the state seed lab was pretty much swamped. As I said, they look at thousands of samples, and many of those samples came in as duplicates this year, where they were sending in both Dynasty and Rancona, trying to figure out which treatments they could use on which seed lots, because not everybody could get the Rancona they wanted, et cetera. So it really created a degree of havoc in the seed industry this spring here in the Southeast. As we look back now, we're starting to sort a little bit of this out. Did Rancona solve all our stand issues this spring? No, it did not. There can be seed quality issues apart from any pathogen-related activity. And when we look at the effect of the pathogens themselves, I teach introductory plant pathology. I pull up the disease triangle that shows the interaction of pathogen, host, and environment. That's what determines the outcome in disease. And this system is one of the more complex systems that we work with, because the pathogens, you have a whole range of pathogens that can attack a peanut seed and seedling. Some of those are seed-borne, some of those are soil-borne, and they'll be different in every field. Then you have the host in there, and the host is not just a peanut plant that's out there growing. It's a seed that's germinating and starting to develop and changing very rapidly metabolically. So the host, what's going on in the host is very complicated and very rapidly changing. And all of that is driven by environment. Environment affects both of those dramatically, and it affects which pathogens can be active. And for example, this spring in May in Georgia, our season would be characterized more as wetter and cooler. We started out hot early, but then it turned, we planted the bulk of our peanuts, temperatures dropped, we had a good bit of rainfall, and that really was not as conducive for Aspergillus to be active as a seedling pathogen as what it could have been. So we may have dodged a little bit of a bullet with the environment we had back in May. Well, the bigger question then, obviously, is why the sudden increase in A. flavus, as well as the difference in seed treatments. And I think that's the important thing we needed to look at to try to help us understand and avoid this issue in the future. I just got done talking about the importance of environment. Environment is hugely important on, as I mentioned, the host, as well as the pathogens. And as I mentioned earlier, A. flavus is favored by hot and dry conditions. I just pulled up a little environmental data from Tifton last year, because what we're looking at this spring is the effect of what happened in the field last year. These peanuts were grown in the field last season. Critical time for development is August, September, October. These are some data from middle of August to the middle of October. These are 2 key months for the maturing and development of the peanut crop last year. And this is the water balance for those 2 months in Tifton, 2016, 17, 18, 19. You can see the degree of water deficit, the degree of stress that these peanuts experienced during those 2 critical months when those seeds were trying to mature. And it was intense. I think I saw peanuts wilt faster last year than I've ever seen them before in my career growing peanuts. It just did not cool off, and it stayed hot, and it would not rain. I was very sympathetic with the Texas farmers this morning and describing their condition, because it's really frustrating when you know how much good a single rain can do and you just can't get it. The other factor there is temperature. These are the similar data. This is a 4-inch soil temperature, so this should be a more buffered type temperature down a little deeper in the soil. And this again is that 2-month period. And you can see the average temperatures there are usually in the low 80s. And this past year we were up there about 84, pushing 85 degrees. That's a big difference in the soil temperature. And again, just reiterates the degree of stress from heat and drought that these peanuts were suffering last year. So in answer to the first question, I would say yes, environment clearly was involved. Well, the other thing from a pathology standpoint, I've been in APRES quite a while. In fact, I gave my first talk at APRES, I forget the year, I think it was 82 or 83, back before many of you were born. But anyhow, be that as it may, my first talk was on fungicide resistance and sclerotinia. And I've continued to deal with resistance ever since. So that's one of the first things that comes to my mind when I see a pathogen that we have been controlling suddenly not be controlled. And we'd been concerned about this with Aspergillus niger. So Brian Jordan is a student here at Tifton working with Dr. Culbreth, and he's been looking at Aspergillus niger and indeed has found some QOI resistance. As I mentioned, that's the ingredient in Dynasty as well as Abound. He has documented that in A. niger isolates from peanut seed here in Georgia. So we knew we were starting to deal with this issue, but A. flavus had not been addressed. So I would like to give some credit here to Dr. Imran Ali, my colleague across the hall who runs our molecular diagnostic lab. I told him about the issue we were having. He was very interested. So we collected a large set of isolates from the seed that I was evaluating, and he was able to do a very rapid assay and also look at the molecular mutations that are involved with resistance. And it was clear, as you can see here, concentrations. This is of azoxystrobin from 0 on the left over to 100 parts per million on the right. The line of plates at the bottom would be a sensitive field isolate, and you can see it's pretty well shut down at anything above 1 part per million. And we had a lot of isolates in there that could grow almost uninhibited up to even 100 parts per million. So it was very evident that fungicide resistance in A. flavus was playing a part in this picture. Just as a reminder, again, the QLI is what we're seeing the resistance to, is the azoxystrobin in Dynasty, and also again then in the Abound that we were spraying in In-Furrow. A little bit of validation. I do field trials each year with seed treatments and In-Furrow sprays. I'd like to credit Keith Rucker with Bayer for taking these pictures. When I do a field trial, I don't put the reps all beside each other in case anyone's wondering. So Keith flew this field with the drone and was able to piece the different reps together. To the far left is the untreated check. So that's peanut seed with no seed treatment. And that's really not atypical. I see that type of stands or even worse without seed treatments. And for a good many years, we could remediate that to a large degree with an Abound In-Furrow treatment. And so here in the center is the 4 reps with the Abound 6 ounces applied In-Furrow, and there was no significant difference in stand, and I have seen that for a couple of years now, actually. And I think clearly now we have the data to show that it's attributed to the resistance that's developing in A. flavus and A. niger. Part of the good news message here today, on the right, we have plots that were treated with vellum total In-Furrow. We've been using that primarily as a nematicide. It's a new class of fungicide. Thankfully, that class of fungicide does have some reasonable activity on Aspergillus. And so it is helping to remediate this seedling disease and problem that we're having and help to accommodate the resistance to the other class of fungicides. So where do we go? What does the future hold? As we look in the crystal ball, I would be the last to say that I know what the future holds, but I will make a couple comments on the things we do know. I think we know that global temperatures are rising, environmental extremes are increasing. NASA tells us that very clearly with data. And I think the guys in West Texas would attest to some of that. I would also say it's another trend we're seeing that's very clear that it impacts this is our use of fungicides. Years ago, when I started my career, we didn't spray that many crops with fungicides, at least row crops. Now we have the things we rotate peanuts with in South Georgia. We've got cotton, we've got corn, sometimes we have soybeans, we have vegetables. We're using more and more fungicides all the time, intensively, on all of these crops. So our fields are being exposed year after year to multiple exposures of these same fungicides. We're putting QOIs on all of these. We're putting azoxystrobin on these. And so we're getting a much greater exposure to fungicide, and that's probably the greatest risk factor there is in developing fungicide resistance is the degree of exposure. And in some cases, we're even spraying QOI fungicides to try to increase yield in some crops, even when there's no pathogens present. And I think the current results that we're getting might tell us that that might not be the most prudent practice. So in summary, try to wrap this up. We do have a pathogen we've dealt with for a long time. We're dealing with that pathogen in the context of a climate that is changing and becoming ever more favorable for the pathogen. We have relied heavily on fungicides to control it. We're seeing that pathogen rapidly adapt to those fungicides and not be controlled, at least to the same degree it used to be. We're dealing with pathogens that are highly seed-borne. I think I showed data to show that. The one aspect I didn't stress, though, is the aspect of spread. And if we have resistance occur in a pathogen, if it's, say it's a soil-borne pathogen that doesn't really have a way to spread, it might not be that big a deal because it's not going to get around. But when you have a pathogen that's airborne, a foliar pathogen, it can spread easily in the air, or in this case, it can spread in the air, but it can also spread in the seed. What better way to spread it than to have a large lot of peanuts, put them in bags as seed, and send them all over the country to be planted. And so we've got, really an ideal mechanism of spread. And I think the handwriting's on the wall that we're going to be dealing with this resistance pretty widespread in the very near future if it's not there already. The other thing then, as I pointed out, is this pathogen falls in that relevant interface of plant health and human health on several fronts. One is the mycotoxins we're all familiar with, and then the fact that it can be a direct pathogen to both humans and plants, and the impact that that may have on our fungicides. As I mentioned, our triazole fungicides are some of the most important we use in plant pathology. They're also used in human medicine. If there's any conflict here between the use of these tools in human medicine or plant medicine, I would speculate that human medicine will win out. So the triazoles are under attack for other reasons already. This could be another nail in the coffin. As I said, these fungicides are critical. I showed you at least one new one, a new active ingredient that does offer hope, but I will say that that class of chemistry is also very susceptible to resistance developing. So we've already lost or losing the QOIs, the triazoles are in danger. If those go, the SDHIs, which is the next class, will be very much endangered as well. So the bottom line, as I see it, we need to find the weak links. I think Diane's presentation was right on target. We need to look at this whole system. We need new approaches to control. We need to pull out the stops to try to undermine how this pathogen works and stop it any way that we can. I will put in a plug here briefly for two talks that are part of this meeting that are very relevant to what I'm talking about here. I've got a paper that gives more detail on the peanut seed treatment changes that I described. There's also one by Dr. Ali then on the molecular characterization of these isolates, demonstrating the fungicide resistance. And he also has some information there on aflatoxin, looking at these strains, the markers for aflatoxin production. Excellent paper. So if you'd like more information, please make it a point to look these papers up, and there may be some others as well. But with that, I think I am about done. I do want to acknowledge these folks who helped with this work, played a big role in it. Thank you for your attention. And I guess if we have any time, Johnny, I'll try to answer a question or two if that's allowed.
Sure. Yeah, we've got a couple of quick questions here. Did you happen to look, does the seed treatment Cruiser Max have any effect on Aspergillus?
I did not have it in this assay, but it would be essentially the same as Dynasty.
Okay.
Because that's the fungicide component is the same.
Okay. Then we have another quick question. How can you tell the how can you tell on the differences, or can you tell on the differences between peanut seedling death with fusarium wilt and A. flavus and A. niger? Can you tell those apart? I guess is what they're asking.
Well, you can. I mean, crown rot is with A. niger is so diagnostic. I mean, 99% of the plants that I see dying in my seedling disease trials are crown rot. And they wilt like that after they come up, and you pull them up and you've got the black sporulation right there on the hypocotyl and the shredded top of the hypocotyl. And it's just, that's A. niger. The others can be a little more tricky. The gold standard pathologist is plating the tissue out and seeing what grows out of the diseased plants or the diseased seedlings. And there can be a range of these symptoms. So sometimes you can recognize it visually, sometimes you have to actually plate them out to tell.
Okay, great. Well, thank you, Dr. Brenneman, for a great talk. A lot of good information in there. And we'll, as we move forward, we have more questions coming in for all the speakers, and hopefully we'll be able to get to those at the end. Dr. Brenneman, would you say that let me make sure I'm on the right one. Would you say that A. flavus resistance to fungicides contributes to carryover and increased aflatoxin problems into next year's crop or the second year's rotation crop?
Yeah, that's a good question. I think almost certainly it would. I mean, we're kind of speculating here, but you know, once you have that fungicide resistance trait in the gene pool, so to speak, you know, it's not going to be responding to the fungicides we're using in the field. It's not responding to the fungicides we use on seed, and you're just going to selectively select for those resistant strains, and they will be the ones multiplying, and they have a selective advantage because they're not responding to the chemicals, and I think they will proliferate throughout our industry.
Oh, yes. Jumping back to Dr. Brenneman, how do you see the benefit of looking for genetic resistance on peanut to Aspergillus as a tool for controlling aflatoxin?
I think it's a great goal. I think, you know, Corley's kind of addressed that and illustrated some of the difficulties and the challenges of actually doing it. It's not an easy target, but I'm a huge fan of genetic resistance. I think it's the best way to control any pathogen if it can be done, but it's not easy to do, as he showed.
Yeah, kind of a moving target. Yeah. Well, is there any indication of a difference in A. flavus incidence in farmer saved seed compared to the commercial seed that you've seen?
There, let me unmute. You know, I'll be honest, I have not, especially with this problem developing so recently and the limitations we had being in the lab this spring and all. In the past, our main problems we have seen, and this has been more with A. niger, has been farmer saved seed. And it often has not had sometimes has not had the degree of care in the field or in processing and storage that commercial seed has. It can also be more concentrated. Seed lots commercially often are mixed, and if you have a lot that has some Aspergillus in it, it gets diluted with other seed, whereas seeds tends to be that one seed source from one field, and if it has it, then you've got it in a high concentration. So our issues have been farmer saved seed over the years.
Okay. Okay, well, great.